Everyone. Thank you. Good afternoon, everyone, and welcome to today's webinar on accelerating biocuration with AI. I am Francisco Azuaje, Director of Bioinformatics at Genomics England, and I'll be your host for this session. We have an engaging hour ahead of with two top talents of Genomics England who will share their knowledge and experience on this subject. First, we will have Arina, who is a scientific curator. And joining us also is Sam, who is an applied machine learning engineer. They are the driving forces in our ongoing work that is demonstrating the value of Gen AI for supporting the analysis of research publications and diagnostic knowledge management at Genomics England. Before we begin, a quick reminder that we will have a question and answer session following the presentation. So please submit your questions or comments through the chat, and we will address as many as possible. Without further delay, over to you, Arina.
Thank you for that introduction, Francisco. So today, just to give you a rough agenda of what we'll be covering, we'll be talking a little bit about genomic testing within the NHS Genomic Medicine Service. We'll then go over curation of gene diseases using published literature, and then how we've kind of used machine learning and text processing in order to find scientific literature. And then I'll be presenting some results of our pipeline and some future directions for the project. So Genomics England is a company that was set up and is owned by the UK Department of Health. So we do many different things in healthcare and research, but one of the company aims is working closely with the NHS to deliver and improve genomic testing in England, which will allow the diagnosis, treatment, and prevention of rare diseases and cancer. So genomic testing within the NHS is done through a service called the Genomic Medicine Service. So a patient with a rare disorder may be referred to this specialist service, at which stage their sample will be taken and sent off for DNA sequencing. So this kind of gives us their unique genetic code that we will use to try and figure out if there are any changes in that patient's DNA that may explain their condition. So hopefully this will allow us to kind of establish a diagnosis in that patient and create a report that is then returned back to the clinician who ordered that test. And they'll be able to kind of explain that result back to the patient. So at Genomics England, we mostly focus on that data analysis part of this process. So in order to determine whether a change in that patient's DNA is responsible for their condition, what we do is compare their genome to something we call the reference genome to pick out any kind of differences in their DNA, from that of this kind of standard DNA sequence. However, every individual carries differences compared to this reference genome at around 4 to 5 million sites. So in most cases, this variation is just part of completely normal differences between human beings. So kind of figuring out which one of those specific changes relates to a rare disorder, we need to be able to understand the kind of impact of different genetic variations. So what we can do is filter these differences in a patient's genome based on a number of different things to kind of whittle it down to the ones that are most likely to be causing a disease. So one of the things that we can look at is looking at genes that have previously been linked to a rare disease that resembles what we can see in the patient. So this kind of increases our chances of being able to make a diagnosis. At Genomics England, we have a resource called PanelApp, where we record evidence relating to genes in human diseases. So PanelApp is a free open access resource, which means that although it is used within the NHS, it is also available to use for the whole scientific community and clinicians from all around the world. So PanelApp contains something that we call gene panels, and this is effectively a list of genes that have been grouped together based on their association with a similar kind of disorder. So in PanelApp, we have over 300 different panels, which relate to a variety of different conditions. So kind of ranging from neurological disorders to metabolic disorders or dermatological kind of conditions. So the range of disorders that we kind of cover is really broad. So on the left and right of the slide, you can see what kind of a simplified representation of a gene panel. So the symbols represent gene names, and these are color-coded based on the strength of evidence that links that gene to that particular disorder. So a green rating signifies the kind of highest level of evidence, meaning that green genes, there is enough evidence for these green genes to use them for the analysis of a patient's genome. Amber and red genes mean that there is maybe some evidence linking that gene to a particular disorder, but it's not quite enough to use them for diagnosis. But this rating system is kind of a dynamic process. So as new evidence emerges, the rating of a gene may change. And that's where the curation team at Genomics England come in. So we will assess any new evidence that emerges and kind of try to ensure that the gene panels on PanelApp are as accurate and as comprehensive with kind of the latest knowledge of disease genetics. So we do use multiple different sources to kind of make an assessment of whether a gene has enough evidence to use for diagnosis or not. But published literature is really kind of a key source of evidence for us. So, for example, you can see with this gene here, maybe previously there was not quite enough evidence for it to be added to the panel. A new publication that we will look at and assess if that kind of contains kind of sufficient amount of evidence for us to then classify that as a diagnostic gene. This may then lead to that gene being promoted and being added to a diagnostic panel. So the new kind of understanding can get applied to real clinical practice. So traditionally, we would find new scientific literature related to gene disease relationships by looking in PubMed or relevant scientific journals, or kind of based on feedback that we would get from experts in the community that would tell us about maybe some interesting papers that have recently been published. But this has been a very manual and therefore time-consuming process. And the kind of corpus of literature is really growing. Not all the papers that are published relate to kind of our field. And when it comes to specifically gene-disease relationships, our understanding is also constantly changing and evolving. So it really has been difficult for us with the kind of manual process that we previously had to stay up to date with all the kind of latest literature. And it was kind of making it unfeasible to keep up with the latest publications. This is kind of what sparked our discussion between the curation team and the machine learning team at Genomics England to kind of see if there was a more efficient and automated way of finding relevant biomedical literature. So at this point, I'll hand over to Sam, who will go into a little bit more detail specifically about the machine learning aspects of the project. Stop sharing.
Cool. Thank you very much, Arina. Share the presentation.
Yes, that looks good.
Brilliant. Cool. So thank you, Arina. That was a very good intro. Text is obviously quite a complicated medium. Like it can take lots of different forms. There can be very high variation in text, especially when we're talking about things like the scientific literature corpus, right? Where we have text that's spanning many different fields of study. There are different types of text. So for instance, review papers. You also have more primary literature sources, books, which are also very different in style. And so coming up with like heuristic or rule-based patterns to be able to deal with these sorts of things isn't very effective. And that's why machine learning has had quite a long history within the text processing world. And there are quite a few different tasks that have been well developed in this field. So for instance, machine translation. So whenever you're using things like Google Translate or DeepL, these are running machine learning models in the background that are taking your text and essentially doing sequence to sequence mapping. So taking your input in one language and then mapping it into a sequence in an output language. There are other things such as information retrieval. So we have a whole body of information and you want to find basically the answer to your question that is in there. And this is quite similar to a more interactive based question answering, right? Where you have a box where you might be inputting text and then the model is also responding to that text with answers. And the pipeline that we're going to be touching on today is sort of taking quite a few different components from these different fields. We did run a proof of concept before this, but this is sort of the final scaled up pipeline that we are running in a production setting, enabling us to deliver papers to the scientific curation team, which then get integrated into their workflows. So we ingest approximately 12,000 papers a week. And if we were to run the pipeline sort of continuously on that, it would take about 6 hours to process. So well within the time limit. And the whole pipeline is essentially trying to take this massive blob of papers and whittle down all the papers that aren't relevant to this particular problem. And so, for instance, we can think about this as having several different steps, filtering and processing based on some heuristics, doing a named entity recognition step. We're then doing some vector search to be able to like find papers that are semantically similar to what we're after. And then the final concluding step is an LLM interpretation stage to actually pull out all the relevant information that we're interested in. And using this, we're finding roughly one positive hit per day. So the filtering and processing step is the first bit. It actually doesn't involve any machine learning, but it is very important and manages to remove, I think, about 60% of the papers that aren't really relevant to us. We remove, for instance, 884 different journals that just are not relevant to gene disease association anyway. And these are things like the Journal of Agricultural Food Chemistry or Frontiers in Veterinary Science. There's never going to be a publication that we care about in this literature. Because Arina and the biocuration team are very much interested in primary literature only, these are like new case studies that have been founded and reported. They aren't particularly interested, for instance, in review articles or books. So these also get culled at this stage. We then do some filtering on the text itself. So, for instance, we remove all the references in the file as well, because these aren't really going to contain any useful information, and just add to the text that ends up having to be processed, slowing down the pipeline, and it also introduces noise that can affect performance. The way we ingest the articles is in an Excel format. So we also do some pre-processing to remove all of the metadata and also all of the XML tags that are present in the text. And this just gives us a very nice, clean, trimmed down final product that our models can deal with more easily. The next step is a named entity recognition step. So named entity recognition is essentially trying to find specific types of word in your article. So typically named entity recognition is thought of as trying to identify things like organizations in a piece of text. So for instance, in a newspaper, or addresses if you're trying to do some de-identification or things like that. In this case, we have a model that is specifically tuned to find genomic entities within the text. And so, for instance, we can see in this piece of text here that the model has highlighted the word ceramide, where it is also followed by the word receptors. So this is indeed a reference to a genomic entity versus the first word in the paragraph, where it is talking more about a material. So it is able to identify the similar word, but in different contexts. We then convert the text into machine readable embeddings, and I'll go into detail in this in a second. But this essentially allows us to do searching of text via semantic meaning rather than having to do keywords, which allows us to cover more of the sort of fluid nature of disease terms. So, for instance, if we think about a particular disease such as or phenotypes such as intellectual disability, this can be referred to in many different ways inside a paper, and we want to be able to capture all of those. And so this approach allows us to pick out all those pieces of information. And so what does this actually look like in practice, right? So words have a lot of different meaning based on their context. So for instance, if we think about the sentence, Sam went to the bank to get money for his holiday, and here's me with approximately 6 million Indonesian rupiah. It was definitely the richest I've ever felt holding that big wad of notes. But we can also think about then the word bank in a slightly different context. So for instance, Sam enjoyed the view from the bank of the river. And so while the text, that individual set of characters, looks identical to a machine, its actual meaning inside that sentence is very different. And we can think about plotting this on, for instance, a number line, where a more negative value is pushing this meaning more towards the value of money, and a more positive value is then, for instance, pushing the value more towards a meaning of river. And then smack bang in the middle is where it has a relationship with neither. And this is a very simplified concept, but is essentially the root of what is going on when we're trying to convert text into a machine readable format known as an embedding. So what we do in practice is we take a chunk of text like this, which has been pulled from a paper. We feed it through a machine learning model that has been trained to do text modeling. And what it is going to do is essentially generate an embedding representation of that model. It is taking all the context, all the meaning of that paper, and it's trying to squeeze it all down into a set of numbers that look approximately like this, a vector. And these particular ones are 1536 by 1 for this one piece of text. And from that, we can then do downstream models, which are able to take that and really pull out that meaning. And we were using the Amazon Titan model, which is available on AWS. So essentially what this looks like is we will take all these different papers, we will chop them up into different chunks, and these chunks are approximately 2,000 characters in length. And we do this because if you think about all the meaning that there is in an entire paper, then you might start to lose a lot of the signal as you're sort of expanding the scope of that. So we really try to chop this up into more manageable sections. But when we retrieve a section, that means that entire paper is likely valuable. So what we will do is we will create a vector store, and this is essentially powered by the FICE module, which was developed by Facebook. And this allows us to essentially hold all these vectors together in memory and then query them. Yep, so there's our batch level vector store. And then here's the interesting bit. We can now take our disorder of interest, we send it through the exact same model, and this time the vector that is produced, the embedding that is produced, essentially is a compressed version of all the meaning of that particular disorder of interest. We can then compare that to all the vectors that are present in our vector store and try to find ones that are particularly similar via, for instance, a cosine similarity. And that will retrieve papers that contain information that is similar to our disorder. And then the final step is to pass the entire model, the entire paper to a large language model for interpretation. And then if the model thinks that the paper is valuable, it is going to extract a whole load of evidence. It is going to structure that evidence, and then we can pass that on to the curation team for final assessment. We've been using Claude for this process, and the reason that we use Claude is that when we kicked off this process, it was the only model that had a context length of, well, I think it had a context length of about 200,000 tokens. So it was the only model at the time that was available that could ingest an entire scientific article. It was also very conveniently available within Bedrock, which is an AWS service, which meant that we could build everything around it within our estate very easily. So this was the first prompt that we were using to query the model. I'm sure by now everyone is familiar with large language models and the way that you prompt them to get answers back. But this was our initial prompt that we went with. So Claude expects everything to be in a human assistant format, which is why we have this structure right here. The opening is often referred to as a system prompt. It is setting the scene. It lets the model know what to sort of expect from the task. And then we inject the paper into the prompt right here. Then we have a few different sections which are essentially trying to moderate the model's output. So for instance, we found, probably because it was a slightly medical seeming task, that Claude could quite often be overly cautious in the interpretation, and we didn't really want that. We wanted, ideally, our experts to be doing that assessment, not the model. So just give us everything you can, right, if it's relevant. We also didn't want the model to be making assumptions about gene names, because gene names change over time. So we also inject all the different names that a gene has been known by. Again, at this point, we didn't have any nice filters in place, so we wanted to ask about secondary information and that sort of thing. And then there's a nice little trick that you can do, which is asking the model to think in thinking XML tags before generating an output. And this worked reasonably well to start with. However, it isn't really an approach to that scales up very well, because all we're outputting at this point is essentially a summary of the evidence, whereas we wanted much more structured output, which allowed the curators to sort of know what they were getting. And so our sort of second iteration of this attempt looks quite different. We are still inserting our paper, but in this case, we're essentially asking the model just to take the paper on faith and just, for every gene that is in that paper, that's mentioned in that paper, pull out the relevant information if it is there. And this means that we can just sort of whole hog do an entire paper in one go, rather than having to iterate over every gene that is present. And then we're asking the model to extract the data in this more structured format. So for instance, give us that gene name, and then give us bits of information that we're particularly interested in. So for instance, the segregation status of that gene disease association, and pull it from a list of values. So for instance, full, partial, or unknown. We don't want any messiness. We don't want the model making its own choices. We're giving it very clear instructions on what to do, and this works remarkably well. Other things we were interested in are, for instance, the sequencing method, because we're more interested in whole genome sequenced approaches rather than things like panel-based approaches. And then also pull out the phenotypic indicators. The source, is it primary, secondary source, or unknown? Because papers will quite often put their discoveries in context and will mention, oh, this gene has previously been shown to be related to a disorder. And again, we're just primarily after that primary literature. And then the biocurators have also found that those summaries that the model have generated been exceptionally valuable. So still give us those summaries, but that gets tacked on at the end. And this is our final architecture that we've built within AWS. We're using full text articles, which are available from the biomedical repository Europe PMC. We pull those in via API. They get stored into an S3 bucket. And then essentially we have the filtering component, the retrieval component, and the interpretation component. So ingest articles, we do our remove irrelevant journals and publication types here in AWS SageMaker. We have a named entity recognition model, which is based on PubMedBERT, which has been fine-tuned to recognize genomic entities. Do our embedding generation, store this on S3. And then finally, this is going into the summary generation and generating the final list of the final table that gets delivered to the curation team for analysis. And at this point, I'll hand back to Arina.
Okay, perfect. So now I want to take it back to how we kind of applied this pipeline that Sam has just talked us through. And kind of how we assessed its performance. So as I previously mentioned, PanelApp contains over 300 panels that are used within the Genomic Medicine Service. And one of those panels is the intellectual disability panel. So looking at the kind of official criteria of how intellectual disability is defined, in the National Genomic Test Directory. It's a moderate, severe, or profound global developmental delay or intellectual disability. So we kind of wanted to just focus on just one disease area at a time, rather than looking at all the genes and all of the disorders that we have in PanelApp. And this was basically to kind of limit the number of papers that we as the curation team needed to look at, and kind of allow us to properly assess the pipeline. So the reason that we chose the intellectual disability panel is because it's the second most applied panel within the Genomic Medicine Service. And it feeds into the pediatric disorders super panel, which is made up of several different panels. Which is actually the most applied. So it's a widely used panel within the NHS. And so it's important for us to be able to keep it as up to date as we can. But that update process can sometimes be a bit complicated. So the reason for this is that intellectual disability disorders are genetically heterogeneous, which means there's lots of different genes that kind of can cause similar looking disorders. There are frequent new discoveries that are made within these disorders. So there's new patients and new genes that are being published frequently. So hopefully that gives us kind of results on a more frequent basis that we can assess. The disorders are also often syndromic, which means that, you know, by focusing on this panel, we would hopefully also be able to look at other disease areas. and as Sam already mentioned, the kind of disease vocabulary that is used in the literature can be quite diverse. So intellectual disability can be reported maybe as cognitive deficits. So it's not as simple sometimes as just searching for intellectual disability in PubMed. So just to kind of, again, take it back through the workflow of how our team worked with the machine learning team. Sam would kind of kick off the pipeline, which he has just explained, to look for new literature relating to intellectual disability. And we were kind of aiming to do this on a roughly weekly basis. Looking for kind of papers that were published within the last week, but this date range could kind of be adjusted as needed. And then this would kind of generate the report, which Sam would check just to make sure that everything was as expected. And this would then be delivered to our team for review. So this is kind of an example of the sort of report that was being delivered to the team for review. So it kind of contains useful information that was contained in that prompt, such as kind of the gene that entry is relating to, the PubMed ID, which kind of would allow us to then check the report against the original publication. And also these kind of custom fields, which we as curators use to assess a gene-disease relationship. So things like segregation, the kind of sequencing method that is used, the kind of clinical presentation of that patient, and kind of to get to this point of having just the most useful information did take a bit of tweaking. So it was definitely an iterative process of us receiving a report, checking it, feeding back to Sam on what was kind of useful and not, until we kind of achieved this desired result of a useful report. So the report also contains a summary, which contains kind of an overview of the key bits of information from that paper. And this was really useful for us to be able to assess the performance of the pipeline. So it kind of allowed us to check whether the relevant and also accurate information was being picked out of the publication. It also kind of allows us as curators to make a preliminary assessment of the paper without having to read the entire text in order to kind of determine whether a paper is likely to be useful or not. So under time constraints, this can kind of allow us to just focus our efforts just on the most useful kind of publications, so kind of saving us time. And overall, we were finding that the summaries were accurate and contained kind of correct information. But sometimes the model kind of lacked domain expertise, which I guess is expected. And we know that this could be addressed by kind of feeding the model with maybe certain definitions of the concept that might not be very obvious. Such as maybe disease penetrance, which sometimes it was making kind of not totally accurate conclusions about, but that's kind of expected. But overall, the summaries contained the correct kind of information. So due to the time constraints, we were not doing a full curation after receiving the report. So what we would do instead is kind of do this quick triage of the prioritized papers and kind of feedback to Sam whether there was kind of useful papers included within the reports, and those that were not were used to kind of maybe refine things and figure out why not the right papers were getting picked up. So papers that were useful were ranked by us in order of priority. So the ones that looked like they might lead to addition of a new green gene, so those are the ones that are used in patient analysis, were given higher priority. And that was added to our curation backlog to get to when we eventually had time to kind of do the full curation. So I just want to go through the kind of results of this whole triage process of just looking at the reports and figuring out if the papers within that were useful or not. So in total, we received 184 gene paper reports. So if a single paper contained more than one gene, that was counted as a separate entry. And this was over 21 total reports. So in February 2024 was when we completed the proof of concept, just to kind of show that our idea was even feasible, which thankfully it was. And this was kind of followed by a period of a few months of completely rebuilding the pipeline based on what we'd learned during the proof of concept project. So we did things like improve the report. We upgraded the LLM from Claude 2.1 to 3. We expanded to looking at more genes because initially we were just looking at genes that were already on the panel. And we also changed the literature repository that we were using from PLOS to Europe PMC. So this just took a little bit of time to kind of set things up in a more production setting where we could regularly start receiving the reports. So we did start receiving the reports on a more regular basis from September 2024. And as I say, we aim to do this about weekly. But as you can see, especially with October, this wasn't always possible. So the gap in October was because we were kind of experiencing some issues with the Europe PMC server. So this kind of meant that they were not putting out any papers. So if they're not putting out papers, that means we're not really able to run the pipeline. But this kind of gap, so when we're not able to run the pipeline, as I mentioned before, we can extend, adjust the time period. So that kind of accounts for this big spike that we got at the beginning of November, because it looked at all the papers that we might have not been picking up that were published in October. So generally speaking, the reports that were delivered, they did consistently prioritize useful papers. So out of the 184 gene paper reports, 101 looked useful from this preliminary check and were actually added to the curation backlog for future review. And each paper did contain at least one useful paper for us to look at. So kind of showed that, we were picking up what we were expecting. Just under half of the papers, we actually found that they were not useful and we decided not to curate them further. And so just to go through some of the reasons for why maybe a paper was prioritized by the pipeline, but was deemed not useful by the curation team. So sometimes the disorder of the patient in the paper did not align with the kind of criteria, so the panel scope for testing within the NHS. So that would be things like mild intellectual disability or neurodegeneration. But it was fine, acceptable for us for these kind of things to get prioritized, as we kind of want that judgment to fall with the curator rather than the model. So it still shows that the pipeline was kind of doing what we were expecting. Some patients also had changes in other genes, so it was not possible for us to conclusively implicate any single gene. And that's just part of genetics. And again, we wouldn't want the model to kind of discard papers based on that. We would again want the curation team to have the final say. There were also, especially towards the beginning of us running the pipeline, quite a few papers that were coming in from these secondary sources. So kind of saying gene X is linked to intellectual disability, but that original paper that it was referencing was already in PanelApp. So these were very rarely useful, and to kind of refine the number of papers that we were getting back and kind of alleviate some of the burden on us, we now have decided to exclude these and no longer include them in our reports. Other things, other reasons that maybe a gene might have been prioritized, but actually was not useful, is the gene might have been reported under a different name. So genes have different aliases, or maybe a locus was picked up as a gene. But in fact, the gene that we're actually interested in was already in PanelApp. So this is kind of an area that was potentially future refinement of the pipeline. And lastly, there were a few papers that were just not relevant for what we are trying to do. So those were things like genome-wide association studies. And again, that's something that we could add for future refinement potentially. So based on the papers that were added to our backlog, we wanted to get an idea of whether the papers looked like they contained useful enough information that it would potentially lead to the addition of a new green gene. So those are the genes that are used in patient analysis. So kind of giving us an idea of whether it's really even worth curating these further. So we did find that it looked like there were about 37 genes, new green genes, that could potentially be added to the intellectual disability panel. And 14 of those looked like they were also relevant to other panels. There were also 17 genes that looked like they applied to other panels, but not intellectual disability. So again, this kind of application was far more further reaching than we initially even intended. So in total, we were predicting that about 54 new green gene associations could be added to PanelApp by running this machine learning pipeline for about 6 months. So it kind of reassured us that we were kind of on the right track. Looking beyond the green genes, the remaining we thought would be added either as amber or red. So although these are not used in patient analysis, they are also important to record because more evidence can come to light in the future, and this publication might be used in combination with that evidence to then potentially promote it to green in the future. So, for example, if a completely new association is found and there's only one family that has ever been identified, we would add that as a new red gene. So just because it's rated red is not an indication that it's necessarily poor quality evidence. So obviously the final step, once we've kind of done the triaging and prioritizing of papers, would be to actually do the full curation of the papers within our backlog and determine whether a rating change on the panel is kind of warranted or not. So I can say we have now actually curated all 101 reports. So these results that I'm showing you is kind of the final outcome of the pipeline from the last few months. So following the full curation, we did have slightly fewer green genes than we initially predicted. So there were actually 48 different new green genes that we added. Most of these are not actually being applied to patients yet, as we do have to go through an NHS approval process before they can be added to the diagnostic panel. But this will be the recommendation of our team to the NHS. So there were slightly more red genes than we anticipated, but this is probably expected. As kind of in the first pass check, you want to add things with the highest rating possible. But actually, when you're actually curating it, you're probably a bit more careful and conservative because our decisions do impact patient analysis, so we want to get it right. But overall, our estimates were pretty accurate, and I can say we definitely did find new diagnostic genes. So that is very exciting. So if we look specifically at the green genes more closely, we wanted to understand why these genes were previously missing from the panels that they were added to. So a significant proportion of those genes were completely new associations. So this is kind of showing that we were able to keep up with the latest discoveries that were being made in our field. A few were already known associations, but they were maybe amber or red genes that previously did not have sufficient evidence. And actually finding the publications the way that we did then kind of led us to better understand those conditions and then led to their promotion to green. The category that we as a curation team have been particularly interested in is these 17 genes, which relate to existing genes that were green on other panels, but were not green on all the panels that were relevant. So this is kind of a known challenge of curation, particularly for those kind of complex disorders with multiple different manifestations, which can be a bit difficult to pick out. And so it kind of shows there is probably some gaps in the coverage of our panels. And we are really interested in kind of exploring this area further to see how we can bridge that gap. Potentially another idea for a future machine learning project, I don't know, but we'll be seeing what we can do to kind of bridge that gap. Okay, so what we wanted to do was compare machine learning to our kind of standard sources of how we would find new green genes. So a feature of PanelApp is a crowdsourcing tool. So experts from the scientific community can kind of log on to PanelApp and suggest papers or changes to the gene panels, which means that clinicians that actually see patients in clinic and use these panels can influence the sort of genes that are being included. We also have the Diagnostic Discovery Genomics England initiative. So this is where findings from patients that go through the genomic medicine service can be fed back into the panels that will be used on patients in the future. So we have always relied on these two sources, so crowdsourcing. So we wanted to compare how machine learning compared for finding new green genes that were missing from the intellectual disability panel. And what we found was that machine learning was more reliable for finding new diagnostic genes. So it was more consistent and more comprehensive at searching all the new published literature. And this makes sense because feedback from expert communities is restricted by their time and their engagement. So if we can use machine learning to kind of get ahead and add new evidence as it emerges in a timely manner, we can hopefully save valuable NHS resources of kind of adding genes as they become evident. So just quickly, I want to go over some of the kind of pros and cons that we found of using ML for finding biomedical literature. So automated literature searches we found were more efficient and comprehensive at checking the kind of latest publications. The Claude LLM was easily kind of adaptable to what we were trying to achieve. So it was good, performed well at mining the scientific literature, and we could tweak it to kind of get the sort of information that we wanted out of it. Text mining was better at kind of finding buried bits of information that can easily be missed as a human curator, especially when you have very busy curation day, when you're looking at one gene after another, it can kind of all become a bit of a blur. So using the tools like this can really help focus you to the relevant bits of information. And again, machine learning can be used to kind of distill that key information from a paper without having to read the whole thing. So kind of saving us a lot of time. So looking at some of the cons, a lot of these are very standard known, I guess, drawbacks of machine learning in general. So our approach is limited to open access publications. So anything behind a paywall, we're not really able to get to. And as you can see with that gap in October, we are reliant on the kind of literature repositories. If papers are not being put out there, then we're not able to kind of apply the pipeline. The output of the AI is limited by the quality of the publication. So if there's not very much information in there, or if it's not great quality, then obviously the output's just not going to be very good. It is difficult to assess what results we have missed. It's not impossible, but given that we have been constrained by time, it's not something that we have been able to really look into. Contradictory evidence may be overlooked, particularly, you want as many genes on the panels as you can that are relevant. So sometimes, there's a bias towards making an association. And in order to achieve the desired result, the pipeline does take a bit of refinement. So that just obviously takes some time. But hopefully once you have designed a pipeline that works, that will kind of save you time in the long run. So to finish, I just want to say that from the standpoint as the curator that's worked on this project, I can say that machine learning has been helpful for kind of scaling our curation process and managing the increasing volume of information, kind of making it more feasible for us to curate new publications and update gene-disease relationships. In the future, so obviously we have shown that the pipeline works in the way that we want it to, but we can kind of apply it in different contexts to see kind of what other information we can kind of gain from it. So that might be accessing all of the literature relating to a gene to kind of give you an overall summary, not just looking at one publication. We can also expand to other disease areas beyond intellectual disability and kind of see how that compares, and expanding to other entities beyond coding genes, so things like non-coding regions. And one thing I do want to kind of finish on, the last point, I guess, is that, machine learning is a very powerful tool, but it is not a replacement for human expertise. And a balance between humans and tools is really needed for us to gain the true benefits from these new tools. So on that, I'd like to thank everyone that's been involved in the project, everyone at Genomics England and the team at AWS who have supported myself and Sam throughout.
Thank you, Arina and Sam. We have time for a couple of questions. Maybe let's start with one from Alice. Super interesting presentation. Did you assess how many papers were discarded by the ML pipeline but were actually useful? And if not, what is your method to mitigate potential false negative classifications? Who wants to take this one?
Yeah, I can give that a go. It's something that we haven't really had enough time to delve in yet, and I think there'll be a bit of a lag before there's enough evidence that we can do this. We'll have to wait for a lot of these papers from other sources to be identified and flagged. So, for instance, those NHS reviews that Arina mentioned, and then we can cross-check that with our pipeline and say, at what point was this falling out of the pipeline and why, and what can we do to mitigate that? But it's not something that we've really prioritized at the moment.
Good, thank you. And we have here one from Eleanor. I'm not sure I understand the question, but I think it's connected to false negative again. Why were the papers that, and connected to the crowdsourcing comparison, I think, why were the papers that were left as expert reviews not all picked up by the machine learning tool?
So I believe it's these kind of these papers that were suggested through other sources. So we are looking just at Europe PMC, which is only one kind of source of literature. So that it could be because those papers just were not in the resource that we were looking at. There was something else. But there were some papers that were identified both by machine learning and experts. So there obviously is overlap. But I'd say so we're obviously only looking in one literature resource, and plus we're only looking for a certain date range, so within the week or the month or whatever, since we started running the pipeline. So the papers that are suggested by experts may have been published in the past beyond that kind of date range that we're looking in. So that could be a reason.
Thank you. Maybe the last question by Mike. Do you know why the genes found by Genomics England curators were not found by the pipeline? And can the pipeline be refined so that the genes concerned will be found next time?
Yeah, so I think that kind of is a similar question. So yeah, I think we could expand the search and also look into those papers that were identified by other routes and kind of see if our assumptions can be confirmed. And if not, then obviously we would want to look into why the pipeline didn't pick those up, but it's not something that we have looked into.
Thank you. Maybe one for Sam from Bernard. Are we at a point where AI can autonomously run the pipeline from end to end, or are we still chaperoning the processes through the pipelines? When can we expect the former to happen?
Yeah, so as it's been developed, like since I haven't made any changes to it in a very long time, it's very self-sufficient. It sort of runs with a cadence, and then those reports pop out at the end and get delivered to the curation team. In terms of iterative refinement, I think it's going to be a little while yet before we're sort of letting the AI do its own refinement on this. Just because there's a lot of scope for it to go wrong, really, and we're not mature enough to be able to sort of automatically be tuning things as we go. There's definitely a lot of scope for improvement, however. For instance, like the disease terms at the moment, we are just using the name of the phenotype rather than like an actual proper formalized definition. So a lot of improvement that can be made.
Terrific. And within the context of a highly regulated clinical level application or like research. Okay, unfortunately, we don't have more time for questions. A couple of people were asking about access, the availability of the code and a paper. We are planning to do that. We are not ready yet. However, I have shared a blog that Arina and Sam previously published with more details. I have added that website to the chat. Please have a look. And thanks everyone for joining us today. The recording will be posted online soon. For anyone who wants to rewatch or share with others, please. Really appreciate your time and the excellent questions. And see you next time.
Thank you.
Thanks, everyone.
Bye.
Bye.